Hello, today's topic is DNA fingerprinting. It's a robust tool for establishing human identity. The concept is very interesting, but I'm afraid it's a bit lengthy. So let's jump into it right away. DNA is a wonder molecule. Nature has used this molecule to create the blueprint of life. All the characteristics that we consciously or unconsciously develop are dictated by the instructions recorded in our DNA. Basically, DNA molecules are long chains of sugar, phosphate and nitrogenous nucleobases arranged in a complex double helical pattern. DNA fingerprinting is a laboratory technique used to establish a link between the two DNA profiles. It's a fascinating science. To understand the basic nature of DNA, we can use the analogy of language. Every language has its own alphabet. DNA has 4. These are nitrogenous nucleobases: adenine, guanine, thymine and cytosine. Their combinations are strictly guided. Adenine always combines with thymine and guanine with cytosine. Arranged in specific order, they form meaningful sequences. In a word, arrangement of letters as well as their size always remains the same. But there may be variations in the length of the spaces inserted between them. Meaningful sequence, which we call genes, actually code for proteins, and they are highly conserved, means their arrangements don't vary. But the spaces sequences can have different lengths. They are a bit variable. DNA fingerprinting is a matching technique where DNA samples from 2 sources are analyzed to see whether 2 DNAs are identical or not. Here the key word is comparison. It's routinely used by FBI to investigate crimes. It's also used in parentage testing and immigration disputes. The basic theory of DNA fingerprinting is like this. Except for identical twins, each person has a unique DNA pattern. If DNA from 2 sources show identical pattern, then either they came from the same person or from his or her identical twin. You know that comparing DNAs is a hell of a job. So do we have to study the entire DNA molecule? Human beings are largely similar. So it's logical that our DNA, which guide our appearance and emotions, will also be similar. In fact, if we write down the DNA codes of 2 random human beings who are no way related to each other and match them in a software like plagiarism detector, we'll get more than 99% match. Only less than 1% will be different, and this different makes us unique. So while comparing DNAs, rather than looking for a match of the similar section, it's much easier to look for the match of some tiny sections where dissimilarities are more common. So here is what we actually do. Some tiny segments of DNA where variations are common are chosen. In the non-coding sections, there are repeated units that vary in length among individuals. One particular type of repeat, known as short tandem repeat, is easily measured and compared between different individuals. These sequences are useful tools in the DNA fingerprinting study. They are used as DNA markers. Tiny spot of DNA where a particular 4 to 5 base pair sequence is repeated at tandem. These are the ones known as STR or short tandem repeat sequences. The STRs are present at specific locations of chromosomes. For example, D7S820 is a marker used in the study. It's found on chromosome number 7. Actually, the base sequence in this marker is GATA, G-A-T-A, guanine, adenine, thymine, adenine. And it shows a tandem repeat pattern. Means this GATA sequence is repeated without any space in between. It will look like GATA, 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 GATA or GATA, 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 GATA. In some individuals, the length of this segment is 20 base pairs, that is 5 repeats, whereas in some other, the segment may be as long as 64 base pair. In fact, these segments can have 12 different lengths of Gata sequence. Each variety is called an allele of the D7S82 marker. For the D7S82 marker, there are 12 alleles. In a pair of homologous chromosomes, there will be 2 alleles of a particular marker. What is the number of possible arrangements of 2 alleles from a pool of 12? To answer this question, I advise you to go to the concept of permutation and combination. If n is the total number of alleles from which k alleles are taken at a time, the number of arrangements where 2 alleles are different is given by nck or n choose k. Here, for the D7S82 marker, the number where 2 alleles are different is 12C2. This is equal to 12 factorial over 12 minus 2 factorial over 2 factorial. The number comes to 66. These 66 markers will have different alleles in them. Add to this the 12 arrangements where both alleles are same. The total number of arrangements is 66 plus 12, that is 78. So the chance of getting the same combination of D7S82 marker in 2 random individuals is only 1 in 78. More the number of markers used in a comparison, less is the chance of finding a match in the study. Now we come to the actual comparison. In the USA, FDA uses the Combined DNA Index System or CODIS. The system simultaneously uses 13 markers. Hence, the probability of getting a match by chance is less than 1 in a trillion. Finally, an example. Look at this table of STR profiles. I have taken it from Wikipedia. Here we have STR profiles of 3 people. First one belongs to a mother, second one of a child, and third one is of a man the child alleges as his father. The first marker is D21S11. The mother has allele number 28 and 30, and the man has allele number 29 and 31. Look at the child's profile. The child has one allele common with the mother, that's 28, and another allele common with the man, that is 31. Similarly, in the second marker also, allele number 10 is common with the mother and allele number 11 is common with the father. For the third marker, allele number 14 is common with the mother, allele number 16 is common with the alleged father. So similarly, for all the markers, one particular allele is common with the mother and another common with the alleged father. So these sort of profiles cannot come out of coincidence. When you take number of markers and match the allele profiles, and if you find 50% of the alleles have come from one parent and the 50% allele has come from another specific individual, the decision is quite simple. The alleged father is the actual father of the child. The take-home message. The DNA fingerprinting is not a genotype analysis. Entire DNAs are not compared. Only some tiny sections where variations are common are extracted and compared. And these sections are known as STS. Next, more the number of markers used in a comparison, less is the chance of finding a match in the study. When 2 STR profiles are found to be identical, we conclude that they belong to the same person. Thank you.